Good morning. Today I am going to talk about for the summary of CLCA process for establishing breakpoints and quality control rates. Okay, the Clinical and Laboratory Standards Institute CLCA is an international voluntary not-for-profit interdisciplinary standards developing and educational organization accredited by American National Standards Institute that develops and promotes the used for consensus developed standards and guidelines within the healthcare community. These consensus standards and guidelines are developed in an open and consensus-seeking forum to cover critical areas of diagnostic testing and patient health care. CLCA is open to anyone for any organization that has interest in diagnostic testing and patient care. Information about CLCA can be found on www.cellsci.org. The CellSA subcommittee on antimicrobial susceptibility testing reviews data from a variety of sources and studies, ex in vitro, pharmacokinetics, pharmacodynamics, and clinical studies. To establish antimicrobial susceptibility test methods, breakpoints, and QC parameters, the details of the data necessary to establish breakpoints, QC parameters, and how the data are presented or evaluation are described in CLCA document N23. Over time, microorganism susceptibility to an antimicrobial agent may decrease, resulting in a lack of clinical efficacy and or safety. In addition, microbiological methods and QC parameters may be refined to ensure more accurate and better performance of susceptibility test methods. Because of this type of change, CLCA continually monitors and updates information it documents. Although CLCA standards and guidelines are developed using the most current information available at the time, the field of science and medicine is always changing. Therefore, standards and guidelines should be used in conjunction with clinical judgment, current knowledge, and clinically relevant laboratory test results to guide patient treatment. Additional information, updates, and changes in the document are found in the meeting summary minutes of subcommittee or antimicrobial susceptibility testing 8 HTTP self-say.org meetings as file resource. Cell-say reference methods versus commercial methods and cell-say versus use food and drug administration viewpoints. Okay, it is important for users of M02, M07, and M100 to recognize that the standard methods described in CLCA documents are reference methods. These methods may be used for routing antimicrobial susceptibility testing or patient isolators. For evaluating commercial devices that will be used in medical laboratories or by drug or device manufacturers for testing new genes or systems. Results generated by reference methods such as doses, load, and CLCA documents may be used by regulatory authorities to evaluate the performance of a commercial susceptibility testing device as part of the approval process. Clearance by a regulatory authority indicates the commercial susceptibility testing device provides susceptibility results that are susceptibility equivalent to results generated using reference methods for the organisms and antimicrobial agents described in the device manufacturer's approved package insert. And well, Cell-site outbreak points might differ from those approved by various regulatory authorities for many reasons, include use for different databases, difference in data interpretation, difference in those used in different parts of the world, and public health policies. Difference also it is because cells say proactively evaluated the need for changing breakpoints, the reasons why breakpoints may change, and the manner in which cells say evaluated data and determine breakpoints are outlined in cells say document N23. Following a decision by CELSA to change an existing breakpoint, regulatory authorities may also review data to determine how changing breakpoints might affect the safety and effectiveness of the antimicrobial agent for the approved indications. If the regulatory authority changes breakpoints, commercial device manufacturers may have to conduct a clinical trial. Some bet the regulatory authority changes breakpoint, commercial device manufacturers may be to a conduct clinical. Some bet the data the regulatory authority and what review and approval for the change reasons a delay of 1 or more years may be needed if a breakpoint and interpretive category change is to be implemented by device manufacturers in the United States. It is acceptable for the laboratories that use Food and Drug Administration (FDA) cleared susceptibility testing device to use to existing FDA breakpoints. Either FDA or CELSA susceptibility breakpoints are acceptable to laboratory accrediting organizations in the United States. Either FDA or susceptibility breakpoints. Okay. Policies in other countries may vary. Each laboratory should check with the manufacturer's issues antimicrobial susceptibility test system for additional information on the breakpoints and interpretive categories used in this system software. Following discussions with appropriate stakeholders, infectious disease and pharmacy practitioners, the pharmacy and therapeutics and infection prevention committees of the medical staff, and the antimicrobial State Washington, newly approved or revised breakpoints may be implemented by laboratories following verification. So say diffusion test breakpoints might be implemented as soon as they are published in 100 if a device includes antimicrobial test concentrations sufficient to allow interpretation of susceptibility and resistance to an agent using the CELSA breakpoints. A laboratory could choose to, after appropriate verification, interpret and report results using CELSA breakpoints. Thank you very much.